The overall goal of this procedure is to generate and purify good quality inositol pyrophosphate. This is accomplished by first using IP6 as a substrate for the IP6 kinase 1 enzyme to generate IP7. Next, the enzymatic reactions are loaded onto a polyacrylamide gel and the band corresponding to IP7 is excised. The IP7 is then isolated by subsequent dehydration-hydration cycles. The final step of the procedure is to confirm the purity of the product and to determine the concentration. Ultimately, results can be obtained that show the biological activity of IP7 through incubation of IP7 with VIP1 kinase and with the IP7 phosphatase DDP1. These reactions generate IP8 and IP6 respectively.
Though this method can provide insight into IP7 signaling, it can also be applied to other systems such as inositol pyrophosphates like IP8 or the IP5 derived PP-IP4.
This protocol begins with the preparation of enzymatic reactions in which IP6 kinase 1 or VIP1 convert IP6 to the pyrophosphorylated isoforms. For this demonstration, histidine-tagged IP6 kinase 1 and GST-tagged VIP1 enzymes were purified from E. coli according to a previously described procedure. To begin, prepare 10 to 20 independent enzymatic reactions containing IP6 and His IP6 kinase 1 or GST VIP1 as described in the written protocol. Adjust the volume to 50 microliters with milli-Q double distilled water. After briefly spinning the tubes, incubate the reactions at 37 degrees Celsius overnight with rotation. Prepare the polyacrylamide gel apparatus using 24 cm long, 18 cm wide glass plates, 1.5 mm wide spacers, and a 16 lane comb. Next, pour a previously prepared gel solution between the pre-casted glass plates at a volume of 50 ml per gel. Insert the comb and let the gel polymerize for 30 to 60 minutes at room temperature. Once the gel has polymerized, transfer the apparatus to the cold room. Pre-run the gel in 1 times TBE for about 30 to 60 minutes at 200 to 300 volts. Meanwhile, add 1 times orange G dye to each reaction. Prepare a standard control sample containing 2 nanomoles of IP6. Following pre-run, wash each well thoroughly with running buffer using a syringe attached to a 21 gauge needle to remove any precipitate. Then load the samples into the gel, avoiding loading the first 2 and last 2 lanes of the gel. Finally, run the gel overnight at 7 mA per gel until the orange G dye band is within the last 10 cm of the bottom of the gel. To begin isolation of IP7, disassemble the gel apparatus and carefully remove 1 glass plate, leaving the gel on the other one. Cut a small portion of the gel from just above the orange G dye band to the bottom, including the IP6 standard and 1 sample lane. Stain the cut portion of the gel with toluidine blue for a few minutes until the inositol pyrophosphate band appears. Meanwhile, place the previously removed glass plate back on top of the gel in order to prevent the unstained gel from drying. Transfer the stained portion of the gel to a destaining solution for a few minutes in order to wash away any excess of toluidine blue. Then, reposition the gel with the unstained gel. The IP7 band should be visible since it runs slightly slower than the IP6 standard. ATP, which runs faster than IP6, should also be visible. Next, use a razor blade to cut the IP7 band on the unstained portion of the gel. Exploit the IP7 migrating position determined with the stained gel to estimate the location of the IP7 band. Put the IP7 band that was cut from the gel into a 15 ml tube and add 10 ml of milliQ double distilled water. Rotate the tubes for 10 minutes at room temperature. Following incubation, discard the liquid by decanting to remove excess TBE and microscopic acrylamide particles. Subsequently, perform 2 dehydration-hydration cycles by first adding 5 ml of 50% methanol to the tube containing the IP7 gel. Rotate the sample at room temperature for 2 hours. Then, transfer the gel slice to a new 15 ml tube containing 5 ml of milliQ double distilled water and rotate at room temperature for 2 hours. Keep the methanol containing tube for further use. Following incubation, repeat the dehydration-hydration cycle once more. To concentrate the eluted IP7, combine the previously added 5 ml of water and 5 ml of 50% methanol. Dry the combined solution using a speed vac heated at 60 degrees Celsius. Once the samples are nearly dry, transfer the remaining liquid to a 1.5 ml centrifuge tube. Spin the tube for 2 minutes at 5,000 rotations per minute. Collect the supernatant, leaving the bottom 20 to 30 microliters since it may contain acrylamide particles. Transfer the sample into a fresh 1.5 ml centrifuge tube. If necessary, continue the drying process using an unheated speed vac. The recovery of IP7 is dramatically reduced if the samples dry completely. Therefore, it is imperative to terminate the drying process when the samples reach the volume of 100 to 300 microliters. Once the isolated IP7 has been recovered, load 2 to 5 microliters of the sample onto a page gel to determine its concentration and purity. Load several dilutions of IP6 as concentration standards and 4 nanomoles of poly-P marker. After running the gel, visualize the inositol pyrophosphate isoforms by staining and destaining the entire gel with toluidine blue solution following the procedure just described. Following toluidine staining, the concentrations can be determined by scanning the gel and comparing the differences in intensity between IP6 and IP7 using imaging software such as ImageJ. The preparative enzymatic conversion of IP6 to IP7 using IP6 kinase 1 and VIP1 enzymes can be easily resolved using page analysis. The loading of IP6 as a size control, together with toluidine blue gel staining, allows for the identification of the pyrophosphorylated derivatives since they run slower depending on the number of phosphate groups present on the inositol ring. The procedure described above resulted in the easy purification of IP7. Furthermore, analysis of the purified inositol pyrophosphate by Page revealed the purity of the IP7. Interestingly, the 1,3-pyrophosphate IP5 isomer of the IP7 product of VIP1 migrates slightly slower than the 5-pyrophosphate IP5 isomer of VIP7 that is generated by IP6 kinase 1. The use of IP6 standards permits an easy quantification of the concentration of the purified IP7. Before using IP7 for further experiments, its biological activity can be assessed. 5-pyrophosphate IP5 is incubated with VIP1 and with the IP7 phosphatase DDP1. Routinely, the purified IP7 is converted to IP8 by VIP1 and to IP6 by DDP1.
Following this procedure, other methods like a prolonged toluene in blue staining can be performed in order to answer additional questions like isolation of IP8 or other phosphorylated inositol isoforms.